Hello and welcome to our section 3 video where we are going to talk about eukaryotic cells. And our learning objectives are to describe the structure of eukaryotic cells, compare animal cells with plant cells, state the role of the plasma membrane, and summarize the functions of the major cell organelles. So just as we did with our prokaryotic cells, we're going to talk about the components of eukaryotic cells, but in a lot more detail. So we know that our eukaryotic cells have membrane-bound organelles, and one of those organelles is a nucleus. So you can see in this nucleus, we are going to have our DNA. Now, when it looks like spaghetti, like it does over here, we call this chromatin, but they can condense and become rod-shaped linear chromosomes. And our other organelles, also known as little organs, would include things like our endoplasmic reticulum, our Golgi apparatus, and chloroplasts and mitochondria, depending on what type of cell it is. So let's first start talking about the plasma membrane. We've mentioned this before, but it serves as the boundary of the cell. So think about that lining that really partitions the cell from its environment, the outer environment, I should say. And this plasma membrane is made up of a phospholipid bilayer with these embedded proteins that are separating the internal contents. So let's say those internal contents are in here or in here from the external environment. And so this really serves as the gatekeeper as far as having control of the passage of organic molecules, ions, water, and oxygen into and out of the cell. For people that have celiac disease, they have an immune response to gluten that really damages microvilli. And so I'm going to show you on these cells here, which we have all of our organelles in here. You can see at the very top they have these microvilli. Now, what are those? They're these finger-like projections, which we could see over here as well, that are going to be found in cells that specialize in absorption, such as our small intestines. We need this there so we can absorb all of the nutrients. Now, in celiac disease, because these microvilli are damaged, they can't absorb the nutrients to the fullest effect, and that can lead to malnutrition, cramping, and diarrhea. Now let's talk about the cytoplasm. This is our cell's entire region between the plasma membrane and its nuclear envelope. So it's all of this in this area here. And it contains a cytosol or gel-like fluid with organelles suspended and its cytoskeleton. So we'll talk about the cytoskeleton some more, but just as a preview, we do have certain filaments like microfilaments and the intermediate filaments, and also some microtubules that serve within the cytoskeleton to kind of create a framework for the cell. And our cytoplasm is going to consist of 70 to 80% water. Our proteins create a semi-solid consistency, so it's almost like a gel type of fluid, and it will contain things like glucose, other simple sugars, polysaccharides, amino acids, nucleic acids, fatty acids, ions, and derivatives of glycerol. So lots of things. It's like a soup with all of these different little ingredients in the broth. Okay, let's talk about the nucleus and the nuclear envelope. As we mentioned before, the nucleus is going to house our DNA and directs synthesis of ribosomes and protein. So within the nucleus, we are going to see that it contains the nucleolus as well as chromatin, which you can see is the spaghetti-like strands here. We also have our nuclear envelope, and that's on the outside here. And this is going to be a double phospholipid bilayer membrane. Think of it as a special plasma membrane, but just for the nucleus itself. And you could see that we've got these spaces here, and these are called nuclear pores, and they help control what's getting in and out of the nucleus. So we have passage of ions, molecules, and RNA between the nucleoplasm, which is going to be more of the fluid-filled area in here, and the cytoplasm, which we would find inside the cell out in this region. So let's talk about chromatin and chromosomes just a little bit more so we understand the distinction. When we have chromosomes, these are structures in the nucleus that are made up of DNA, meaning it's hereditary material. And all of our eukaryotic organisms have a specific number of chromosomes. For instance, we have 46 chromosomes in each of our cells, whereas fruit flies only have 8 chromosomes in each of their cells. And to change the number of chromosomes for the organism will not be compatible with life. Now, yes, sometimes we can have 47 or sometimes we can have 45, and you may live quite a while, but it's still going to have effects on the body that we will talk more about in pathophysiology. In chromatin, this is when we have our chromosome basically unwound. So you can see it all along in here, and it just looks like a bunch of threads. We are going to have protein in here, and we could see an image of it over here, like the histones that makes up the complex of it. And we'll see this during growth and maintenance phases, which we'll get into later on. Let's talk more about that nucleolus that we saw over here. This is going to stain a very dark purple, and we'll see aggregates of ribosomal RNA with their associated proteins to assemble the ribosomal subunits that are then going to be transported out of the pore of this nucleus and into the cytoplasm so that we can go through different processes like translation, for instance, in which we are going to read some of our genetic information in order to create proteins. So those ribosomes are very important in allowing us to create proteins that we use throughout our cell and throughout our body. And our ribosomes, just to go into a bit more detail, are pictured over here. And this is the genetic material I was talking about, which is our messenger RNA. The ribosome reads it and then can basically take the codon here and translate that into an amino acid strand. So we would call this a peptide, and lots of peptides can come together to eventually make a protein. More information on this in chapter 5. And this may be also bound to our rough ER. That's how it gets its name, rough, and we'll talk more about this in the coming slides as well. Or in our intracellular plasma membrane or outer membrane of our nuclear envelope, or they could just be free ribosomes and just be living in the cytoplasm. And you could see they're made up of 2 subunits. We have our large subunit over here and its small subunit. And we'll find this in just about every cell in our body, but we'll find that we have an abundance of them in our cells that make lots of proteins. For example, like our pancreas, that would be making lots of digestive enzymes, which enzymes are a protein. Now for our mitochondria. You may have learned that this is called the powerhouse of the cell. They have little they're basically little energy factories within our cell, and you could see that they have a oval shape to them and has an inner membrane and an outer membrane. The inner membrane is kind of folded into what we call cisterna that contain enzymes for cellular respiration reactions, and I believe it's chapter 6 where we'll talk more about these reactions. And the area that's surrounded by the folds will be called the matrix. So this area in here would be our mitochondrial matrix. They also have their own ribosomes and DNA, and we believe that they might have been a prokaryote a long, long time ago and were basically consumed by a eukaryotic cell. So they do have their own DNA and they are able to reproduce. And so through some of their respiration reactions, they're going to be able to convert chemical energy stored in the glucose molecules into chemical energy, which will be stored in ATP, also known as adenosine triphosphate. This is essentially our energy currency inside of the cell. So we call these processes cellular respiration when we use up our oxygen and we give off CO2, carbon dioxide. And of course, we breathe out that carbon dioxide when we exhale. And this is where that carbon dioxide is coming from. And we will find mitochondria in almost every cell as well. And there are certain cells that need a lot more energy, like our muscle cells, in order to fuel contraction. Next, we have our peroxisomes, and these are small round organelles that are enclosed by a single membrane. I accidentally put an S there, single membrane, and they're going to carry out oxidation reactions that break down fatty acids and amino acids. So they will do this by releasing hydrogen peroxide, which can damage the cells, but in peroxisomes, the enzymes break it down into oxygen and water. So we have some peroxisomes within our liver that can help to detoxify alcohol, also other poisons. We have glycoxysomes, which serve as the peroxisomes in plants, and they're responsible for converting stored fats into sugars. You may also hear this term vesicle or vacuole quite often. These are membrane-bound sacs that function in transport and storage. So think of them as like the cars or the buses of the cell. And the vacuoles are going to be larger than our vesicles. And the vesicle membranes can fuse with either a plasma membrane, okay, out here, or other membrane systems in the cell. So we're going to see this when we talk about the Golgi apparatus, for instance, when we're able to take something like a protein and put it in a vesicle and ship it somewhere else in the cell or out of the cell. And some of the vacuoles we'll see will break down macromolecules such as lipids. Now let's talk about animal cells versus plant cells. We'll see in animal cells we'll have microtubule organizing centers, and our plant cells will have the same. In our animal cells, we'll have something called centrioles. We see them pictured over here, and they are going to help with our process of mitosis in order to basically copy our cells. And we'll also have our centrosomes, which is what we're going to call all of these. I mean, by all of them, I mean the two centrioles that we have here. We'll talk about this more on the next slide. And then we've got lysosomes as well, which will help digest certain waste products or even pathogens within our cell. And within our plant cell, we have a cell wall. We don't have a cell wall within our animal cells. And our chloroplasts, which are kind of like the mitochondria of the plant cell, and other specialized plastids here. And we have large vacuole, which we mentioned can help with the breakdown of macromolecules. So let's come back to that centrosome. So this entire thing is called a centrosome. You could see that we've got these microtubules, and they also will extend microtubules to eventually attach to our chromosomes during mitosis. And so each one of these are called a centriole. And you could see that they have a cylinder of these 9 triplets. So 1, 2, 3, and you could see they're separated out here of these microtubules. And the complete function is actually not known, because if a cell has a centrosome removed, it can still divide, and plants don't have centrosomes and can still divide. And then we have our lysosomes. Like I said, we've got some digestive enzymes in here, so it really serves as a garbage disposal. And enzymes are going to aid in the breakdown of proteins, polysaccharides, lipids, nucleic acids, and worn organelles. Now, we don't have lysosomes in our plant cells. Our plant cells instead will have their digestive processes take place in the vacuoles. And these lysosomes will be active at a much lower pH than the cytoplasms, meaning it is going to function best in an acidic pH. Now let's talk about the cell wall of a plant cell. This is going to be external to the plasma membrane, and it serves as more of a rigid covering that's going to protect the cell, and it helps give structural support and shape to the cell. Now we'll find these in plants. We mentioned that we'll also find it in bacteria and fungi and some protist cells. We know in bacteria, they also have within their cell wall a special, special molecules that create a peptidoglycan. Remember I said we could have this peptidoglycan be really thick or really thin. Just as a preview for microbiology, gram-positive bacteria will have a thick peptidoglycan wall, and gram-negative bacteria will have a thin peptidoglycan wall. And we kind of use that to our advantage to help identify them and also see what certain antibiotics work best on these different types of bacteria. And then we have cellulose in our plants. And we said our chloroplasts are kind of like the mitochondria. So they too have their own DNA and ribosomes. And these chloroplasts are going to help carry out photosynthesis. We know that plants are autotrophs, which means they're able to make their own food, whereas animals are heterotrophs and they have to ingest their own food. So we've all heard about the process of photosynthesis. It's going to be a series of reactions in which we're going to use the carbon dioxide, water, and light energy that we get from the sun to make glucose and oxygen, which they are going to use that glucose for energy and release the oxygen. We have inner and outer membranes on the chloroplasts. You could see the inner membrane here and then the outer membrane here. And the space inside has a set of interconnected and stacked fluid-filled membrane sacs that are called thylakoids. And we can call each of these stacks granum, and the fluid-filled area is known as the stroma. So individually, thylakoid, altogether granum, and the fluid here is the stroma. We'll also see that we have a pigment in here known as chlorophyll, and this is a green pigment that's going to capture that light energy to help drive the reactions within photosynthesis. Now let's talk about that large vacuole in our plant cells. This has an important role in regulating our cells' concentration of water in a changing environment. So if it loses water, the plant is going to wilt because that central vacuole shrinks and the cell wall therefore is unsupported. So its role is to support that cell's expansion when more water is present.